synaptic membrane here. Okay, so you got your neurotransmitters going on. Cute little guys floating around in there. I am going to try my best to remember. This is where it starts to get a little bit more complicated. This is why I was excited to be able to do this lecture, to see if I can do this. Okay, so this one is going to be bound by 1. So we'll make a little docking site right there. Okay. Now, this is called a G-coupled protein receptor. Okay. So what does G-coupled have anything to do with it? The G is actually for guanine. Okay. So here you have these 3 proteins that are all connected. I just realized I made Mickey Mouse there. All right, cool. So one section is the alpha, okay? And then one section is the beta, and then the gamma, okay? Greek, get your Greek symbols here, okay? And where G protein comes into play is guanine, okay? It has to do with guanine, okay? So you have you probably heard of ATP, ADP, and AMP, okay? Adenosine triphosphate, diphosphate, or phosphate. You also have the same structure as far as the phosphates go, but with GDP, diphosphate, okay? Now the diphosphate is connected to this alpha here, okay? So I'll just say that, actually, I should probably make this one a different color so we can keep track of what's going on. All right, so if I have, boom, this is my GDP, okay? So if I get, you know what, before I go on, I'm gonna check my camera again. Sorry guys. The fun of being new to this stuff. Okay, we're still good. So a neurotransmitter will bind, okay, and it will cause what is called a condensation reaction to occur. When I say condensation reaction, that's where we're getting into biochemistry, okay? So this is actually going to become guanine triphosphate, okay? So we add a phosphate group. Condensation reaction, really quick without getting too much into it, okay, you're creating a connection of different molecules and it's going to release a water molecule, okay, H2O. You wonder why you have so much water coming off of your body all the time. It's because of condensation reactions like this. They occur all over in your body, not just here. Okay, so this will become guanine triphosphate, okay, and what that does is, again, this is where it goes into bunches of different ways that these things can actually work, okay? So we have another protein over here. Now, either this will cause, again, getting into biochemistry, a separation of the alpha subunit and then the beta gamma, okay? So really quick down here, you can have an alpha subunit by itself, or you also have the beta gamma unit that's together. Typically, you're going to be working with the alpha subunit, I believe, okay? But it can be the beta gamma as well, okay? So this will cause a pushing, and this alpha subunit all of a sudden is going to detach, okay, through and connect, okay? So here's your alpha subunit, okay? And boom, it's left there. It goes and attaches to that, okay? And this all of a sudden is receives in whatever ion needs to be created, okay? So now this is activating as the second half of this, okay? Remember I was saying this one serves two purposes, to receive the neurotransmitter and then also as a channel. This one's receiving the neurotransmitter, but the channel is a different protein on the post-synaptic membrane, okay? All right. I want to make sure that I got that good. Let me look at this really quick, okay? Let's see. Should be noted there are 5 different classes of the muscarinic receptor, and they do different types of things based upon where they are. Okay, so in the heart you have these receptors also, except for in the heart you're working with potassium, okay, from the vagus nerve, and it's actually going to slow the heart rate down, okay. So yeah, different types of muscarinic receptors will do certain things, okay? So there's M1 through M5 for different ones. And again, those work with acetylcholine. Let me erase these. It's amazing looking at all the things that are going on in your body all the time. I sit and wonder how we've discovered all of these things. We've discovered a lot of these things very recently actually, with the invention of the electron microscope and different tools we've developed to be able to actually see these things. In fact, those ligand, the snare protein that I talked about, that was actually, I don't know when it was discovered, but the person that discovered it was awarded the Nobel Prize in 2013, I think. So a lot of these things are pretty recent. They're not that old. As far as discovery goes. Okay, we do need to talk about, so I kind of talked about the neurotransmitter does need to go back. It can't just stay in that synaptic cleft forever. Unless you're on those drugs, all right, that I talked about before, then it'll stay a lot longer. Okay. Again, while there are lots of neurotransmitters, in the case of acetylcholine, which is used like I was saying, a lot, you're going to have these structures, these little proteins that come out of the postsynaptic membrane, okay? And these are called, let's see if I can spell this, acetylcholine esterase, one of those nice fun big words, okay? I talked about this in a video a while back, I think. If it ends in A's, you're working with an enzyme, okay? An enzyme is a specific protein that is going to have a binding site and it's going to pull and push to cause different things to happen, okay? And in this case, when we're working with acetylcholine, remember how I said acetylcholine is actually made of acetate and choline? Okay, so let's imagine you got acetate, and then the other half of the structure is choline here. This acetylcholinesterase is going to break that apart, okay? And then when it's broken apart, it's able to be taken back into the presynaptic membrane there, or into the presynaptic terminal, or the baton. And the various mechanisms, it's going to be rejoined together, okay, and it's going to reform acetylcholine, okay. So things are always being recycled in the body and going through those processes, okay. Let's see. Then we also need to talk about monoamine neurotransmitters, okay. Acetylcholine is going to be your main one, okay, that you're going to be working with. Actually, thinking about it, a good way to put it, acetylcholine is going to be in the peripheral nervous system, and then your monoamines are going to be more in your central nervous system. Don't take me for absolute truth, that's always how it is. I swear there's so many different ways and so many different exceptions. I can't really guarantee, but generally that's what it's going to be, okay? So, monoamine neurotransmitters. This here. Monoamine neurotransmitter. Okay, now there's a lot of them, but your main ones that you'll probably hear about, norepinephrine. Now, you're probably thinking norepinephrine, is that the same thing as epinephrine? They're very, very similar. 85% is epinephrine, 15% is norepinephrine. I might have those backwards actually, don't quote me. One's 85, one's 15. Okay. Now, epinephrine is both a hormone and a neurotransmitter, actually. So if it's in the blood, okay, and it's been secreted into the blood, then it's going to be a hormone. If it's in the synaptic cleft, then it's a neurotransmitter. But it's the exact same structure, okay? It's the same thing. It just is depending upon what its function is at the time. You're also going to have dopamine, serotonin, okay? It's probably the big ones you're used to hearing about. So, norepinephrine and dopamine are made from the amino acid tyrosine. Okay. And serotonin, actually, I think it's tryptophan. Don't quote me on this. And I'm actually not sure how to spell tryptophan. You've probably heard of that one from chicken Thanksgivings. All right? Makes you tired. Serotonin comes from tryptophan. Okay. Here. Now, these act, as far as the mechanism goes, across the postsynaptic cleft. It works. Shoot, I probably should have put this over there. Maybe I'll just kneel down so I can leave those up there for a few minutes, okay? Nope, I'm not going to be able to. This is probably going to be definitely one of the more complicated mechanisms. So, I'll start at the first again. So you got your presynaptic membrane. I'll do it a little bigger this time. And then your postsynaptic membrane. Okay. Then, alright, I'll do a little docking one. Okay. You got your snare proteins there. Okay. This is called exocytosis, by the way. Okay, when it's bringing something out. If something's bringing something outward, it's exocytosis. If it's bringing something in, endocytosis. Okay, so you would say that these synaptic vessels are undergoing exocytosis of the neurotransmitter. So this goes in. We're going to do a little protein structure here. And then I'm also going to need one over here. Okay, so this is also going to be using the G protein structure. Okay, so you got your alpha, beta, gamma. Then you got your, your GDP that's binded to the alpha subunit. Okay, so if neurotransmitter comes down and binds, okay, binds to it, you got a neurotransmitter and the synaptic cleft everywhere. Binds to GTP, you undergo a condensation reaction, okay, and that creates GTP. Again, so this part is the exact same thing, okay? Still a G protein coupled receptor, just with an exception here that we'll talk about in a sec. And then, again, it's going to cause the alpha to break away and attach. Now, oh man, I am hoping I'm remembering this. This protein structure here, adenylene cyclase. The cyclase part is right. I might have the adenylene wrong. So don't quote me on that. Look into that and be sure, okay? Don't take my word. But something cyclase there. What that is going to do is turn ATP, adenosine triphosphate, which you've probably heard of, at the energy currency made in the mitochondria, powerhouse of the cell. And that is going to change it to what's called cyclic AMP, okay? This cyclic AMP is then going to act on what we call protein kinase A. Now, remember when I was talking about if it ends in A's, you're working with an enzyme, okay? Kinase, and you're saying kinase, you're going to be dealing with phosphorylation. So what is phosphorylation? phosphorylation is when you're adding a phosphate group. Simple enough, okay? And then dephosphorylation, you'd be taking it away. In this case, we're phosphorylating. So protein kinase A is then going to phosphorylate various proteins, and then depending upon the function that needs to occur, that will be the whatever protein that the phosphate is being added to. Okay, that specific protein will then go and do whatever needs to occur. Again, that's getting into, there's so many different types of mechanisms here. Okay, so as far as what that protein does, it just depends upon exactly what the purpose is and where that is in the body. Okay, so this is called the secondary messenger system. Okay, that is an important word to know. Okay. Secondary messenger system. So your first messenger is the neurotransmitter that activated that, okay? And then your second messenger is actually the cyclic AMP, okay? And then if you're going backwards, I can't remember the whole process of going backwards because obviously this neurotransmitter can't stay in there forever. This is going to be dephosphorylated by an enzyme called phosphodiesterase. Okay, and then this one, phosphatase, will then deactivate this one, not phosphate, phosphatase. Okay, that will deactivate protein kinase A. Okay, which will then, alpha subunit will then come back here, break apart, and go back to this G-structured protein unit. Okay, that is the secondary messenger system. Let me look through this again, make sure I'm still on track. Another thing actually that we do, so when the neurotransmitter is being taken back up here, okay, when it's being brought back, reprocessed, you're gonna have what are called MAOs, okay, and that stands for monoamine oxidase. So monoamine oxidase will deactivate the neurotransmitter when it goes back in by oxidizing it. Okay, you probably heard of reduction and oxidation. Okay, so reducing, you're adding a hydrogen to it. For those of you that aren't too familiar with chemistry, it probably sounds confusing. Why is it reduced when you're adding something to it? I encourage you to go look into it. Get into biochemistry. Okay, and oxidation is when you are taking something with the hydrogens from it. Okay, so you're removing. So these monoamines, the neurotransmitters, they have hydrogens on them. Okay, part of its structure. I don't remember exactly how they're all made up, but it's going to have some carbons there and hydrogens coming off. Okay, but to oxidize it is to take off some of those hydrogens. And there's an expression we have that form follows function, okay, when it comes to physiology and cellular biology even, okay? And so the function of the neurotransmitter is going to change if its form is changed. So when you take off hydrogen, you're going to change its ability to do what it needs to do, okay? And there are different drugs that will actually ruin these monoamine oxidases that will stop them from working, okay? So they can't break apart these neurotransmitters. I can't recall at the top of my head what those are, so I'm not going to try and make a guess, okay? Really quick, let's do one down here. I haven't really drawn down here yet. I kind of talked about inhibition, but I haven't really gone into it, okay? So you got your... A really important neurotransmitter to know is GABA. You probably heard of that, okay? What does GABA even stand for? Let's see if I can... GABA, I want to say guanine, albuterol, Yeah, I can't remember. It's a big word, okay? But, all right, so GABA. You probably heard of it with, like, to help you sleep. I actually took it once a long time ago as an exercise supplement to help me get better sleep. I don't know what that has to do with sleep, actually. But anyways, this, if you got your receptor down here, your GABA receptor, this will bind to it. And the difference with this is going to be an inhibition, okay? So what's going to happen is rather than sodium, you're going to have chlorine. Okay, now sodium is positive, so you're creating that depolarization. Chlorine is a negative ion. So that's when you're going to cause that hyperpolarization, okay? And that is when you're getting into your muscles, okay, if you need that to be deactivated, okay? There's also glycine also that does deactivation, except glycine is a lot more on the central nervous system, I believe, okay? All right, I think that's it. I'm trying to make sure that I didn't actually miss anything. Okay, so again, this is a lot more of the biochemistry of it. It's kind of fun, actually. It's really awesome, right? I think next time we're going to start, now that we've looked, so if you realize I'm kind of starting from the smaller units and working outwards, okay, rather than going the other way, except for the general broad picture of the systems. So in the next video, we're going to start the central nervous system, and we're going to we're going to start looking at the brain and go over some anatomy and see how the brain functions. All right. With that, I think that's it. I think I'm going to close.